Thank you again very much. Great talk by Chiara, and I like to stay in the label free. So bringing you to the Muller matrix and to something that you know very well, probably better than me, that is a phasor analysis application in different ways you can use it. My start is with this picture, just because with this plateau in the cavern, we use light now to try to decipher what's going on outside. Is not so easy. We can see things on a screen, and then we can try to figure out how things outside organize and function. We use light, as all of us in Genoa, in my group, and we try to take advantage of any kind of interaction between light and the sample. The main, the focus of my research and our research is to try to understand something related to chromatin DNA structure and function. That is rather complicated, but at the very same time is a kind of El Dorado for microscopists, because our aim, our idea is to be able to have the very same instrument to follow events at different spatial scales. And I will be back on this later. The object we are interested in this case chromatin, but other macromolecules, have the problem that they are transparent, so you could perform some perfect imaging in transmission, but in order to get details, you have to find something different than shining light and collecting a signal. So you are like in front of this kind of object, a very nice transparent glass, and then you want to decorate it. You have also other methods in order to get contrast, for example, phase contrast, of course, but then you have some compromises between spatial resolution and the ability of understanding what's going on in the three-dimensional shape of your sample. So fluorescence is a very good way for getting information about your system using light. And as we have seen in the past days, including this morning, yesterday, and all the other days, adding a touch that is using multimodal optical microscopy and then performing some image correlation when you sense, when you're able to detect the interaction between light and the matter. So when I was explaining what we had in mind to Marta Fioravanti, that is a graphical designer, she decided to provide us with this graphic, with this picture. You have some ideas, you shine your light, color light, visible light. You want to decipher what's going on in the system. And then we will see that also maybe we need some more computation than we had in the past, including the phasor from our point of view. This rather complicated architecture is only to tell you that according to what we have seen in the last days, we can pack in the very same instrument a lot of possibilities in terms of getting information from our samples. So from selective plane illumination to multiphoton, STED or single molecule, label-free Muller matrix, we will talk about it. We have new detectors like the one discussed by Giuseppe Viccidomini yesterday. So this incredible ability of getting information from single photon producing one electron by time. And so today we are able to build an instrument that is not complicated, but takes advantage of everything we want to, we are able to do. From my point of view, a Genoese or like a Scottish guy, this means that we can exploit the very same light source for producing so many signals that we have, that we can detect. When we use this instrument at different level of organization in our lab, we can try to decipher what's going on in terms of chromatin organization using fluorescence in different systems. So we can play with oncogene that switch on some aggressive event in the cell. We like working on these progeria, including expansion microscopy, in order to decipher why these changes can affect the function in the nest in the cell. We want to study, and this is one point we are very interested, the neuroblastoma, since this affects children and There is knowledge, but not very precise knowledge on this, as on many other aspects in biology. And then, if we watch forward, not backward, you can find this paper on the bioarchives by the Hell group. You can discuss, and you listened yesterday to the MinFlux and to other methods for improving resolution. But today you know that probably you have no more limits in terms of localizing with precision molecules. Here they claim and they demonstrate 1 angstrom in terms of localization precision and 1 nanometer in terms of spatial resolution. So it's amazing. Chromatin again, it's complicated, is compacted. I'm interested in what happens in the interface. And if you have a look here, you immediately, but you know better than me probably, what's going on. I mean, so you have this filament that is highly compacted in the nucleus. And then over the years, we are trying to understand which is the relationship between organization and function. And every 5 years, more or less, our view changes. I read this paper in 2017 using a method that demonstrated how the packed 30 nanometer fiber is something that we have to discuss better. And we have to think about an organization made by 5 24 nanometer diameter chains for a more flexible and fast working of chromatin DNA inside the nucleus. So things are changing every 5 years, our touch, our understanding. Now, my point is, can we perform together some experiments using some reporter that tells you that your cell is in the normal or pathological cell, and check if these reporters, P53, for example, they change their diffusion when you're using fluorescent molecule or expressing green fluorescent protein with respect to what happens when you have nothing inside. Fluorescence is great. I love it. I love all the part related to autofluorescence, of course. But when you add something in this crowded environment, then I think that we have to think more what we are doing. This is the reason why in this window we are interested in the interface and thinking about an experiment that tells us which is the progression of chromatin organization related to a different function in the cell that can go to tumor malignancy or back to normal. Here, we want to try to understand. The situation is overcrowded enough. For this reason, this overcrowding, there's so many molecules, Irene's pushes to say, can we find something label-free, really label-free here? Low dose of light, possibly. Well, if you go to the coffee machine, you immediately realize that what Irene was mentioning with her hand is something that you do with the coffee machine. You insert the coin in a polarized way. And so we decided to use polarized light to insert the coin, the electromagnetic field in the system, in a polarized way, checking better what's going on without labeling the sample. Many years ago, I started paying attention to this and considering the fact that I could detect some chromatin changes. I will be back on this, and not only with this slide, but just to remember when I started on this topic in 91, demonstrating the ability of detecting organizational changes of chromatin, in this case in solution, detected using a label-free approach that we will see right now. So I have the sample, I shine light, I have a lot of physical parameters that I consider: phase, direction, wavelength, intensity. Polarization is something that I think can be of interest. Again, you can put your coin in a polarized way, 90 degrees, you don't get any coffee. If you throw your coin with random orientation in the space, you can get a coffee, not immediately, but you can get something. So we shine light to the sample, and we try to be something that I'm not, so ordered guys. Ordered guise means that we describe the using a polarization vector, different precise polarization states. So unpolarized, any polarization, polarized vertical or horizontal, this S1, S2, 45 degrees, minus 45 degrees, intensity vector rotating right, intensity vector rotating left, so circularly right polarized light. Now, if you describe this with this vector, and you assume that you have this vector as input, you can collect light in a very same ordered way. If in the middle you place a sample, if there are changes between the input vector and the output vector, responsibilities of the object. And if you want to link them, since they are 4 elements vector, well, you can use a 16 elements matrix. This is the Muller matrix. What is nice is that if you combine the different elements, you can work on the superposition of the effect of each of these elements, known polarizers, retarders, any kind of elements you can classify as optical element, plus your sample that is the only unknown. If you play this game, you can try to extract some information that can be interesting. So the way light is depolarized, if you have kind of dichroism, if you have birefringence, so if you have a different behavior in terms of variation of refractive index. So really you can extract a lot of information playing with this kind of approach. And Lou-Chipman approach, so is an approach for extracting dichroism, the attenuation of birefringence, and depolarization from your data. This 16 elements Muller matrix has elements that are defined by the composition of light that is impinging the system and the light you are detecting using a certain setup. So you can think that you have a polarized state generator as input in your system, and then you have your detector, and then you have a polarized state analyzer. And you can work on this in the way you want. What is nice here is that if you use something that I love, that is a photoelastic modulator. A photoelastic modulator is a simple bar of quartz that you mechanically stress at the resonant frequency of the quartz, and at 50 kilohertz, you can get all the polarization states if you shine unpolarized light in. Great. So you can generate all the states and you know them when they are in that states in time. Of course, it's not so, I mean, it's easy, but then of course you have to perform a lot of checks during the production of this light in terms of retardance or whatever it is. But then if you play this game, you have in your hands something generating different states. And so you generate different states, you collect the different compositions, so you build the Muller matrix. You have 16 images, you don't know exactly, which is the reason why you have contrast or not in some of these images. But for example, one image related to plus minus or linear and vertical polarization tells you, like X2 here, something about birefringence, the attenuation. Other elements we can discuss which is the meaning. But what is nice is that when you have a chiral sample, you have one element on. That is the last one of the very first row with some redundancy in the 16 elements. Chirality makes the light having a different behavior with the sample when you shine circularly right or left polarized. So in order to check chirality, or in order to check how dense are the helices or the super helices in the sample, you can play your game having your sample not transparent using fluorescence, or you can try to use something that you know already very well. probably most of you also know the Muller matrix. You have in your lab circular dichroism instruments. With the circular dichroism, you exploit a differential absorption of circularly polarized light. If you go out from the absorption bands, many years ago, someone, Carlos Bustamante is one of them in his doctoral thesis, someone discovered in the group of Ignacio Tinoco, Marcos Mestre, Dorman, that you had a signal, a differential signal, when you shine circularly right and left light to your sample outside the absorption band. Now, if you perform a check change in the wavelength, as they did, they realized that this was due to the scattered light. This means that you can use the scattered light, the differentially scattered light, to get some contrast, so a differential behavior with your light that you can detect with your detector when you shine circularly right or left. And this is called SIDS. I'm more interested in this element, but also the other elements are of interest. Now, the problem I have is that, for example, I can try to set up a system like this one. Again, I'm from Genoa. I don't want to waste too many photos. I have a very high cost, as you have when you have a titanium sapphire laser. You can perform great fluorescence excitation, but then you can also use this red light, as we demonstrated in the paper I've shown you at the very beginning. Using also the red light, I can use titanium sapphire under two-photon excitation, and I can use part of the beam for inducing some scattering, differential scattering effect. This is what we can do. So we start with the titanium sapphire, we go into the sample, we try to check and control very well polarization. This is our polarization state generator. We go through our photoelastic modulator that allows us to have a different timing, different polarization states. And then we can detect the fluorescence signal and a label-free signal. And you could say, come on, but now you are using fluorescence. First of all, I could also exploit second harmonic, third harmonic, and autofluorescence, but now I don't mind about this. It's true. Now I want to use, at the very same time, what will be the label-free signal only when I remove fluorescence and the fluorescence signal, because I need to learn something more about the kind of interaction I'm detecting when I'm in the label-free. So what I do is to shine light and to collect fluorescence on one arm of my instrument and collecting scattered signal on the other line. Now, it's very fascinating the chance of, since we have a modulation and we have an angular or phase variation, we decided in different ways to try to move from the Lou-Chipman decomposition that is very interesting and powerful to an attempt of building a phasor. Now, the problem we have is that we are not sure of what we are detecting. So we're for sure, more or less, we are detecting something that we modulate. And we can follow modulation effects, so the different state of polarization and the different way the system is interacting. But for sure, we need to be more precise. I will touch this later. And so what we did was using some polystyrene microspheres as a reference recently, and some progeria cells. We tried to find a phenomenological way to get different information when you have a pathology or not pathology. What you see here on this phasor quadrant is the average signal from different region of interest in order to see if we are able to detect, to find a space where you can have normal, the blue, and the pathological, and some reference that we can take into account. The signal we are collecting, I'm convinced is a dirty signal now, but we can clean this up. But this is not the initial point now. Again, what we did, using in this case Lou Chipman decomposition, was to try to study with a different setup that is, but it is a setup not using in this case fluorescence, but only using by a reflective mirror the signal coming from the sample in a label-free configuration. And in this case, our polarization state detection is made of 4 different detectors that are collecting changes in the 4 elements of the Stokes vector. And in this case, we got interesting results. For example, in the development of the zebrafish embryo during time. It's nice. We don't need any labeling in this case. And we found also that, sorry for these rotated images, the only way to have this in this picture, we could also have some information about in the tail, for example, of the zebrafish, of orientation of some fibers. Now, we played this game also with second harmonic generation in the past, but this is a more difficult task in terms of Muller metrics. You need really more elements. So this only to say that we can get a contrast that we are trying to understand better today. For this reason, we are trying to find a way for representing the changes with the phasor or Lou Chipman or other methods in case, in order to try to be able to connect with the different cases. It's pure phenomenological from my point of view, as many others works also that are used in medicine. And we want to try to understand better what's going on. Again, in this case, what we do is, if in case we want to perform the phasor analysis, what we can do is consider pixel by pixel what we are doing for all the different Stokes vector we are collecting. And in this case, we can have a representation that, from my point of view, is great, because what can I do? I can find in the phasor quadrant, I can find the position of my horizontal polarizer, my vertical polarizer, the 45 degree, in the very same way when I try to find the lifetime M variation for a lifetime due to having more than one molecule or more than one lifetime packed together. And what I'm interested in is the part, the RCP, that is related to circularly polarized light. We are performing a lot of experiments like this with adhesive stripes that induce birefringence in order to see the effect of noise and so on. But we think that this is very promising if we are able to find, also with your help all together, a key for classifying things in terms of what's going on. This is another case obtained using collagen fibers. Sorry for being fast here. I'm sorry because it was too long, so I want to recover a little bit. There is another aspect of interest. Sorry for mixing the different aspect analysis and the system we are using. In order to generate polarized light, we are also enthusiastically fascinated by the possibility of generating using a Zeeman effect laser. This means that we are able to produce, using our helium neon laser, circularly right and left polarized light without the need of other elements inside, like the photoelastic modulator. But just different attempts to do this. So different possible setup. In order to build this setup with the Zeeman effect laser, we spent definitely less. 300 euros we bought on eBay the laser and the magnets. But now there is the last point. And the last point is nice what we can do. If we use the phasor, we can also continue in our phenomenological approach and we got a lot of results that tell us that using this desk, we can recognize pathologies. But we want to understand something about our chiral sample. And so we have a lot of, let's say, theoretical approaches that we are trying in order to better understand what does it mean when our sample is complicated by a very high density in the nucleus. And we demonstrated different wavelengths with different ratio between the wavelength and the size we are interested in, so the resolution, that we could follow changes in chromatin organization. I want to show you this also because Wasim Ashraf also had in mind that we could try to detect something in the air droplets in case there is coronavirus. We are all very interested in this. And he demonstrated that we have a chance of detecting something and maybe using a phasor approach to have something that can tell us if you have nothing in the droplet or you have something viral that we can classify. We need a library and we need more experiments in order to confirm this. But only to say that this is a very, for us, label-free plus finding a clever way for detecting and extracting the signal is very promising. Because again, we are in this rather complicated short period of the whole lifetime of the cell. I don't know if you ever considered the time here, but probably yes, and better than me. And the high density you have there. And we want to understand which are the mechanisms of compaction and decompaction to drive out through the nuclear pores the information. Now, what we are starting now, and I'm going to be at the end, sorry for me, a little bit longer than expected, also by myself, is that now we continue using fluorescence to compare fluorescence labeling, so density through the intensity of chromatin organization, with the Muller matrix element that is called SID, circular intensity differential scattering, related to the element that is sensitive to chiral organization. The architecture, so, is complicated. We can have a lot of fluorescence approaches, better than the one I've shown you with the DAPI distribution, to be coupled with a label-free. But now, what we need is to switch from this multimodal approach to an intelligent multimodal approach. And we think, and I need also your help, and I think that you also discuss about this in these days, we need some artificial intelligence. So Lisa Cunio, that is a young postdoc in our lab, started working on this data that need to be structured. And we want to reduce the complexity of the data. And phasor is a way for reducing the complexity when we inspect what's going on. Sorry, I am going to skip this, the ICA. I only want to show you what we want to do now, what we are doing now. What we are interested now is to collect a couple of images, fluorescence and Muller matrix, label-free. Selecting the element is not so easy. And after this training of our network, the expectation is that if at the end I will insert only the label-free data, I will have as output the fluorescence image. That is something that I can decipher better and compare better with our data coming from the super resolution and from other methods, including lifetime and all the other methods. So the point is that we want to use Muller matrix approaches for extracting information in an efficient way, put all this data in an artificial intelligence network in order to be able to understand better what's going on in our sample. So the system is rather complicated. It's not so complicated. I mean, it's complicated, but just because you put all the things together, wants to collect the different data and to take advantage of this mathematical approach to get more information from this data.